All right. Welcome, everyone. So this is lecture 13 on computational systems therapeutics. Today we have a guest lecturer, Saathvik Kannan. And I just want to sort of, put the context in which we're seeing all this. So basically, we are in the last stretch of module 3. So basically, module 1 was all about genomics, epigenomics, single cell networks, and circuitry. This is about understanding the workings of the cell, understanding the circuitry of the cell, and then being able to use that to identify specific targets, specific pathways, specific cell types where we should be intervening. Module 2 is all about understanding protein structure. It's about understanding the language of protein function, if you wish, and then knowing where and how to intervene. In other words, from module 1, we have the circuitry. We know what pathways and genes we should be targeting. From protein 2, we're getting closer to the actual structural underpinnings of biological function. Module 3 is all about the therapeutics, all about the chemistry, understanding the landscape of chemical structure and being able to apply that. And we've already seen an introduction to drug development, to do small molecules, and then the representation of small molecules on Tuesday using graph neural networks, transformers, etc. Today, we're talking about systems level therapeutics. What does that mean? That basically means understanding not just one molecule at a time, but understanding the entire landscape of chemical structure. So using the latest in generative AI and latent representation learning, etc., to understand thousands of molecules, millions of molecules at a time, and then how they fit together. And then today we're going to be also looking at agentic workflows, and we're going to look at latent space representation cartography. So basically interactive ways to study those latent spaces. To complete module 3 on the chemistry and the therapeutics, we're going to be looking at causality. And the way to get at causality is through the genetic association studies. So we're going to be looking at disease association mapping, genetics, genome-wide association studies, and disease circuitry of how do we go from genetic regions to individual causal variants to function. And then we're going to look at quantitative trait mapping. This ties in with the electronic health records of module 4, of basically understanding now not just genetic variation across individuals, but also phenotypic variation across individuals. And then the last 2 parts after the quiz on comparative genomics and phylogenomics to understand the underpinnings of disease by understanding the language of DNA and specifically the evolutionary conserved parts. And then we'll wrap up module 4 with modeling metabolism. Okay? Everybody here so far? So that's sort of the context in which today's lecture is happening. And of course, the frontiers of the field are going to be you guys presenting on the awesome final presentations that you have. So this is the bigger picture of systems therapeutics. So why do we call it systems therapeutics? Because it's not about one drug at a time. It's not about finding a needle in the haystack. It's about understanding the cartography. So Saathvik is going to talk about the building blocks of representation learning by inputting the biological data across chemistry, across proteins, and of course, across the scientific literature, but also patents and other representations of language. Then, how do we build representations for these different types of objects? How do we create a language for protein structure by masking individual letters? How do we create a language for chemistry, building these graph structural encodings? How do we build chemical language models? And how do we encode all of that in lower dimensional representations? So basically, how do you build vector embeddings from these different types of data types? Then he's going to introduce contrastive learning for how do we actually build a joint landscape between chemistry and proteins. That basically means that, yes, chemistry can live in its own landscape, protein structure can live in its own landscape, but it is that intersection between the two that really allows us to get at a unified view of biology. And that's, I think, a lot of where the field is going, of basically not having individual machine learning models for a task, but having foundation models. and not just having a single foundation model for one data type, but having these multimodal foundation models that allow you to connect across all of these different data modalities and translate across these different data modalities, and also include human language in there so that we humans can get insights about these latent structures. So Saathvik is going to talk about contrastive learning as a general methodology for learning these joint representations. And then he's going to present his own work on building affinitynet, the first foundation model for understanding chemistry and protein structure and protein function together. These joint representations allow us to go in two different ways. The first is directly visualizing and interacting with these latent embeddings using what we call cognitive cartography and platforms that we have built in our lab for having this human interactivity where you can actually go directly into these latent embedding representations. And then number two, it allows the agents to interact with these latent representations directly. So instead of reasoning about individual atoms, the agents can now reason about higher order representations. And that reasoning can be much more powerful, as we've seen in many different ways in chemistry, protein structure, image analysis, and so on and so forth. So basically, these higher order representations is where a lot of the reasoning happens. Okay? And then lastly, how do we actually bring back experimental feedback from all of these systems? Okay? So today we're going to be learning about all of these things and how we can use these joint representations and these agentic frameworks to interact with basically everything we've learned in the class so far and everything that's ahead. On one hand, the gene regulatory networks of Module 1, the transcriptomics and the circuitry, the epigenomic information, the electronic health record information, and of course, the therapeutics. So without further ado, Saathvik.
All right. So what I want to emphasize more of today is more of the how than the what, because we can see what's going on, but the conceptual underpinnings of many of these are what's actually sometimes more fascinating, especially when we try to go from some of this multimodal data types on the left to trying to make insight out of them. And that's what really today's lecture is about, trying to gain insight from vast amounts of high dimensional data that biology is usually in. So we're going to start with representation learning. Professor Alm has already sort of gone through the underpinnings of representations for small molecules and proteins. But in essence, what's going on here is we have a lot of data types that are there in reality, right? We can have pictures, we can have audio, and at the end of the day, to actually be able to use them in a downstream task, to be able to predict, for example, what is in this picture, we need to convert them into numbers. And there's a variety of ways of doing this, and we'll get into that in a few minutes. But essentially what we want to do is this process here. We want to be able to convert this data into a type of data that is essentially something that we can make inference on, right? And this isn't applicable to just images or audio. It's also applicable to therapeutics and proteins, right? We can go from small molecules to vector representations of them, right? And this will be useful for predicting properties of them. For example, if we could figure out that this molecule is toxic, or we could figure out that this molecule targets a specific protein that we're interested in. And we can do the same for proteins, predicting protein function using these vector representations. But at the end of the day, this demands an important question, right? How good are these representations? How can we make them better? And how can we fine-tune them to the right purpose, right? These representations matter a lot if we want to do inference on them. If we want to learn from them, these representations need to be able to capture the meaning of each of these data modalities. So sort of the first way that we got into representations of chemistry is through fingerprints. And fingerprints are actually quite simple to understand. They're quite easier than what you might have seen in previous lectures, because all they're doing is they're trying to capture the structural properties of molecules. For example, the Morgan fingerprint is one of the most famous molecular fingerprints, and it's a 2,000-bit vector. So it's 2,000 entry points, and it's a bit vector, so it either has 0 or 1, and it signifies the presence of a functional group. For example, we can see this particular functional group is present here, so it gets a 1 at that location. But the lack of a functional group also gives us information, so that's a 0. And we're able to do this iteratively. So the bigger the molecule, you would imagine you could have more functional groups. So we can go across the entire molecule and have all these functional groups. And functional groups determine what molecules are able to do, right? At the end of the day, if you have a specific functional group, that might make it bind more to a specific protein, or a specific functional group could cause toxicity, right? And we're able to capture this through the Morgan fingerprint. This was created in the 1960s, and it still outperforms some of the recent AI achievements. But the motivating question that I have for you today is, at least in the first part of today's lecture, is can we let AI learn its own representation of these molecules? Sure, the Morgan fingerprint is great, but what if AI can learn its own representation of these molecules? Would that be superior? Could that help us to understand some more important aspects of this? So we'll dive a little deeper into this, right? So there are multiple ways we can approach this problem, right? We first need to figure out a way to give an initial representation of this small molecule. So something that you are all familiar with to varying degrees is graph structures. So what we can say is we can put together a molecule graph. So the same molecule, if we take this molecule here, we could represent it as a graph where each node is an atom and every edge represents a bond between these nodes, right? And this conveys enough information that we could possibly take out and do message passing, which essentially conveys some of the information about neighboring nodes through the edges to the other nodes. And through that, maybe we can capture some essential information about the molecule that we could use for then downstream tasks, right? But then there's another way. We could actually combine both of these. We could use this graph as an input to a variational autoencoder, which takes this molecule, encodes it, and we compress it to a low-dimensional representation. And hopefully this low-dimensional representation has carried through many of the major parts of the structure of this molecule. And then what we can do is use a decoder in the same variational autoencoder to then reconstruct the molecule. So the aim is that this low-dimensional representation in the middle might have captured some core properties of the molecule that we can then use for downstream tasks. So this is a proxy for the molecule structure. And that's what we'll keep coming back to. How can we use these representations for then inferencing later on? And this is something that we have seen in the past use this and get great results as well. Now, another way that scientists have approached this problem of representing small molecules is using language models. So you have seen protein language models in the past, right? Protein language models, what they do is they mask a certain amino acid token and they attempt to predict that amino acid. And what's implicitly taking place is, oh, we're understanding the grammar, we're understanding the syntax of protein sequences. But we can do the same for small molecules as well. Small molecules follow a syntax called SMILES, which Professor Alma has talked about. And what we can do is we can sort of mask out certain parts of these SMILES and then say, well, predict what comes there. And using that prediction, we can form a representation sort of similar to the protein language models. And using that representation, it comes back to the core principle which we've been talking about, and that is doing downstream inference, right? We can try to predict clinical toxicity, blood-brain barrier permeance, and so many of these important characteristics of molecules that we want to capture. And it's the same intuition that was there for protein language models, the fact that we are able to capture what is the next token or what is this masked token that applies here as well. So now we sort of inverted this, but the proteins follow the exact same process, right? This is a model called PROT-T5, which essentially follows the exact same process. We mask out certain amino acids and then attempt to predict what that amino acid is. And the hope is that it has learned the core aspects of what governs the grammar of protein sequences. And when we're able to do that, we're able to capture some of the most important aspects of protein structure. And what I find very fascinating is that these protein embeddings actually capture some very meaningful implementations as well. For example, you can see that in these protein sequence embeddings, if we were to convert them down to a two-dimensional representation, which we will get to in a few slides, we're able to see that they cluster by known family. So if we take proteins whose families we know, and then we take them and put them into the embeddings, These protein sequence language models, which have essentially learned the grammar of proteins, are then able to cluster these proteins based on the family. And what that tells us is that machine learning models, which have learned to predict the tokens, have implicitly learned the grammar of protein language, and therefore what protein structure they have also gotten as well. And this is the same philosophy behind other protein models as well. So now this gets at the core of today's presentation, right? We have small molecules like this, therapeutics, ideally thousands of these, millions of these even. And we've got proteins here, protein sequences or structure, and we can numerically convert them into vectors using the processes that I just shared, right? We can do representation learning to figure out what is the ideal representation of small molecules. We could use the Morgan fingerprint. For proteins, we could use protein language models, protein structure. And then we now have these vector representations. And this is where we can say, let's go on to the machine learning. What can we do? But the problem is these were learned separately. So if we want to look at these in a picture, ideally these are in thousands of dimensions, but if we were to look at them in 2 dimensions, what you'll see is that the proteins form an island, right? All of the proteins are in a separate space. All of the drugs are in a separate space. And sometimes this picture is even deceiving because the fact is that these proteins are in probably a different dimension than these drugs. So you can't even put them in the same coordinate plane. So what I'm telling you is that this slide is even wrong in principle because these proteins can't share the same space. But how can we make them share the same space? The principles that we've learned so far also allow us to do that, putting the proteins and the drugs in the same place. And now we have a lot of options, right? We can have what determines where the drugs go in the protein space, or if we were to take the proteins and put them onto the drug space, what determines that? We now have a lot of creativity and flexibility with how we can merge these spaces. So we have 2 problems now, right? 1, how are we going to merge this? And 2, if we merge it, what is the way in which we merge it? Do we want the drugs to go next to the proteins? Which drugs do we want to get next to what proteins? So we've got a lot of complexities here, and that's what we're going to uncover in the next few slides. So the idea behind contrastive learning, and we'll get to that in a second. So I don't actually want to talk about contrastive learning right now, but what I want to talk about is these sort of pairs, right? These proteins are in probably 1,000 dimensions, and for now we're going to imagine that the drug is in 500 dimensions, right? So 2 different dimensional spaces, but again, these are arbitrary dimensions. What we want in an ideal vector space, right, is that the target for a drug, EGFR for example, and erlotinib, which is a drug for this target, should be closer together in this ideal vector space. Do we all agree on this? Okay. We want these closer together in this ideal vector space. But then, if we have EGFR and a drug like ibuprofen, do we want them together? No, right? We want them farther apart in this ideal vector space. So how do we enforce this type of rules that we've put together? So the idea is that we want to attract similar pairs, and here I've got another dot, which I'll come to in a subsequent slide, but essentially the idea is that we want the blue dot and the orange dot to be coming closer together, and this blue dot and this orange dot to be farther apart, explicitly because this blue and orange are pairs, right? This particular drug inhibits this protein, and this drug does nothing to do with this protein, so we want them to be farther apart in this latent space. So now the idea is to enforce this. And I'm actually going to come back to the slide in a second, but the idea to solve this problem is contrastive learning. And this is a strategy that was introduced to us by our friends at OpenAI. And what they actually said was, well, if we have text, like Pepper the Aussie pup, for example, and we have an image like this puppy that's called this, we want them to be next to each other in this ideal latent space, right? So if we take a matrix, right, with pairs that we know that should be together, for example, this particular text annotation should be next to this particular image, right? We encode this using some arbitrary representation learning method. We encode this image using some representation learning method, and we'd have some layers to make sure they're in the same dimension. And the idea is that this vector here, and just to show you an image, is that the image vector for this puppy should be very close to the text vector here. So this is a simple dot product, right? Like if you normalize these vectors, the cosine similarity here should be very high, right? Along the diagonal, all of these numbers should be 1 because the vectors are so close to each other. But if they're in the off diagonal, that means this is an image and text that don't go together. They're not in the same place. Like this is a pair that's not supposed to be paired together. That's like EGFR and ibuprofen, for example. They should be far apart. So their cosine similarity ideally should be closer to 0. Right? Does that make sense? Okay. Now, this is the idea behind contrastive learning. We want to form this matrix that sort of has 1s along the diagonal and 0s elsewhere. And this becomes sort of like your loss function, right? This is your error. This is your ideal expected value. And then you have what you're getting in your matrix. And the subtraction of that ideally becomes your loss function for training a contrastive learning model. Now I'll come back to the previous slide that we were looking at. And contrastive learning can be done at different places, right? And it can be done at different places because if we come back here, this text encoder could come way before. This contrastive learning step can take place at different places in your neural network architecture. So that's the idea between this early fusion versus late fusion steps that we're talking about, right? It can happen at different points in your architecture, and that allows you to do some downstream learning. So if you have different objectives in your machine learning model, you can integrate them with contrastive learning. So you could do it after an attention step. You could do it before an attention step. So it's very much modular. That's the idea that we're trying to convey with this slide. So contrastive learning, when applied to biological data, can take up many forms. But today we're talking about therapeutics. So the EGFR erlotinib example is very clear also here, right? Except they didn't use EGFR and erlotinib. There's a paper by Professor Berger's lab here at MIT. And what they've shown here is that without contrastive learning, if we take this protein here, for example, it's called VGFR2, and these blue dots here, you can see these blue dots, right? Those ideally should be right next to the protein they inhibit. So if the drugs are right next to the protein they inhibit, the contrastive learning that's the idea of contrastive learning. And we can see that happens when they use this model called complex, which is one of the first models to do contrastive learning on protein ligand data sets. And you can see precisely how that works. So the same image and text example sort of applies here, right? We have the protein, and all of the drugs that bind to that protein are right next to that protein in this space. And they've got some cool examples here where it works quite well to predict which proteins and drugs bind together. Now, we're going to sort of look at all of these principles that we've learned, representation learning, joint representation learning, and how we can place contrastive learnings at different parts and see how we took that in our lab to sort of learn the chemical function through their structure alone. So ideally, what we want is to have each of these dots represents a drug, right? And we want these drugs to be forming islands, right? So all of these drugs inhibit protein A. All of these drugs here bind to protein B, bind to protein C, and bind to protein D. So we want this grouping of chemicals by function. And once we do this, eventually want to get to something like this, right? Where drugs are grouped by indication. So you can see here that, again, these are 11,000 drugs. So we'll get to how we made these 1,000-dimensional vectors into 2 dimensions in a second. But essentially, we have these drugs, 11,000 drugs. And what you're seeing is that they form islands. And these islands are grouped by the indication. So all of the drugs in this green island are those for cancer and related diseases. And we can say the same for this blue island, orange island, etc. Right? We want to group molecules by their function. And that happens through their structure. So how exactly do we achieve this? Well, what you're going to see is exactly the principles that we've been talking about come into action right here. And feel free to interrupt me with questions at any point. So right here, we have 4 different modalities. So we've been talking about drug and protein a lot, but the same contrastive learning, as you might imagine, it's just we're putting pairs together, right? That pairs can happen for multiple modalities. So I want to group together the drug and the patent that drug goes with. I want to group together the drug and the protein that they go with. And this contrastive learning can apply there. So the input to our affinitynet model that we've been working on that sort of combines all of this is the drug, the protein, the domains which are in that protein. So the different families that are in that protein, and eventually the patents that cover both those drugs and proteins, right? And then this is where we see that.
That's about the dimensionality. Why so many dimensions?
Yeah. So what you're seeing here is the Morgan fingerprint, which we talked about.
I'm just going to fix the audio. Talk to them.
Yeah. So for the drugs, what you're seeing is the Morgan fingerprint, which we talked about in like slide 4. And what you saw there was that it's a 2000 dimensional vector. And then the proteins are the Prot-T5, like the masked language model that we were referring to. That was the 1000 dimensions, 1024. And then we've got the domains. Those are done using a model that's just a plain language model that can allow us, they mask certain words, and then it allows them to sort of figure out the meaning and grammar of words. So plain language model there for extracting details from domains, because domains are things like polymerases or ligases, and we want to understand the meaning of that. So that's why we use domain. And then the patents are also words, right? So that we use the same model here. Now, what's important to notice about this is that all of these are in different dimensions. They live in different vector spaces. So we need to be able to combine them in a unique way. And the principle behind machine learning is that we can convert from different vector spaces to another vector space. So this is why we have a 1000-dimensional vector that we've transformed them to. And this projector that we have here converts this 2000-dimensional vector to a 1000-dimensional vector. And what do you notice here? All of the dimensions are the same. And this is what allows us to sort of go up and do the contrastive learning, which we'll see in the next slide in more detail. But what you're seeing here, essentially, is that we have now put them together in the same vector space. And that allows us to do a lot of computation in the next slide.
You need to learn a reverse function, or how do you go from that joint latent space to each of the modalities?
Back? Well, yeah, you can't go back using just the deeper projector. But what you can do is build in a decoder, which allows us to sort of learn a reverse function. So right now, natively, we don't have a reverse function, right? Because algebraically, that's just not possible to go in reverse. Yeah.
Sorry, what was the projector again?
Yeah. So it's a great question. So it's just a set of MLP layers. So we've got a fully connected layer, and then some other just ReLU activation function, and then drop out. So it's not anything too complicated. It's just a couple of neural network layers stacked together just to make this linear map as strong as possible. Yes, it has to be learned. Yeah, that's a very good question. And these deeper projectors are what allows us to do the contrastive learning that you're going to see. And the sort of constraints we place on it, you'll see in the next slide. And that's maybe we should just go to that next slide and come back here. Yeah.
Is there just one?
Well, no. They're 3 separate towers. Do you see what I mean?
Oh yeah, but there's just one of these. So where's the residual coming from? Because the previous layer is a different dimension.
Which one? Here?
Yeah.
It's going from the 2000. Well, each one will be defined appropriately.
No, but I mean, like, how do you add the residual if the previous layer was 24 gig? If this one is 1024.
Yeah. So each one has its own. Do you see what I mean? Like if you have the residual layer will come before we convert to the 1000 dimensions. So like it's in the last few layers that the 1000-dimensional conversion happens. So it'll stick to that 2000 dimensions until the very last few layers where so like it will have a residual.
The last layer.
Yeah.
Thanks.
Yeah.
Can you repeat for the class?
Yeah. So essentially what Professor Alm was asking is residual layers, what they do is they take the input and they allow the model to sort of remind itself of what the input is. And that allows it to capture some important meaning if it was lost along this axis, right? What Professor Alm was asking is, well, if it's in 1000 dimensions, how can you add 2000 dimensions to 1000 dimensions, right? And the idea is that the 1000-dimensional conversion sort of happens towards the end. So it still sticks at 2000 dimensions, so we're still able to add, and at the very end, we convert it to 1000 dimensions. So that's the same deeper projector sort of applies for all of these. And the important thing to note is that each linear map is different. It has to be, right? So the 2000 and 1000 here is a different model than the one for protein, than the one for domain, than the one for patent. So we have 4 separate ones that are defined. It can't be the same one, otherwise you can't give a different vector input, right? I'm going to come to the contrastive and then come back to this because I feel like that would be more useful. So we're focusing right on this step here. Don't worry about the second half of the model for now. We're only looking at the first half, okay? The idea is the same. We were trying to bring together the drugs and proteins that are the ones that bind to each other. And we want to also bring in the patents. How do we want to bring in the patents? Well, we want to put the patents in the middle of the drug and protein interaction. So if the drug is here and the protein is here, we want the patent to be right in the middle. And if the representation was learned properly, what we should see is that the patent should be right on top, right? Because the drug and protein are in the same spot. Now, I want to emphasize this again because this can get a little confusing. So we want the drug and protein to be at the same location at this latent space. And once we have the drug and protein in the same place, or at the same time as this drug and protein are entering into the same place, we want the patent to also be at the same place. How do we do that? Well, the drug and protein in the middle is where the patent will go. So ideally, if they're at the same place, the average is just the same as their location, right? So we want the patent to be in the middle. Does that make sense? Okay. And we want to repel the dissimilar pairs per the usual. So how do we actually do this? Well, there are multiple objectives which we use, and I'll come back to this after we go through the rest. But the contrastive learning part of this says that at that point in the model, right, at the point at which we are learning just the linear map from 2,000 dimensions to 1,000 dimensions, we want 2 functions here that enforce the latent structure. The sequence and drug should be put together. And at the same time, we know the midpoint, right? We can just compute the midpoint. That becomes like the image, right? The midpoint becomes the first part. And then you also have the patent, which should be at the same location. Yeah. Yeah, so that's a great question. So the domain, we're not actually doing any contrastive learning on it. Yeah, thank you for asking that. So the domain, we're not going to do any contrastive learning on. We're just going to sort of use as a method to predict affinity, which is the essential aim of this model. So yeah, great question. So now that we've sort of understood the contrastive learning objective here, I'm going to go through the rest of the model, and then we'll come back to the objectives. Again, I'll re-emphasize the contrastive part because I think that is the core. So then once we have these drug, protein, domain, and patents in the same vector space, we can do, this comes back to the early late fusion, right? This is very modular. Contrastive learning can happen at different parts of this architecture. So we can then apply attention. So what we can say is this drug vector has been learned, this protein and domain vector have been learned, and this domain vector, again, we're not enforcing any contrastive learning, it's just a pure linear map for the sake of it. No objectives enforced. So the query is our drug, and then the protein and domain become our key value. And then now we get a cross-attended drug. And we don't, as you can see, use the patent. The patent is only used during the training to enforce this latent structure. We're caring a lot about this vector space that we're creating. But the patent is not used for actually predicting the affinity. The reason we are making this model is to predict the affinity between a drug and protein, right? And we'll come back to how this fared later in the process. But essentially after this, we can predict the affinity of the drug and protein with exceptional accuracy. Now, the question becomes is if we're having a model that can predict the affinity, we can also do some cool stuff with that, right? We can say, well, I'm going to look at this drug latent here. Forget about the first inputs for now. We can look at this drug latent and make that our variable x, and make the predicted affinity our variable y. Imagine we want to maximize y, given that we know the protein target, the drug can change. What we can do is we can do gradient ascent. So you're used to gradient descent, which is about minimizing a function. And now what we can do is maximize a function, maximizing the affinity that we've gotten. And when we do that, well, we've got another problem. Professor Kellis a few moments ago was hinting at this, that saying that can we do the reverse? Can we go from drug latent back to drug Morgan fingerprint? We can't, unless we explicitly make a decoder for that. And that's what we did here. So we went and made in a decoder, and this is trained as part of the model that can reconstruct the drug. So this slightly could remind you of a variational autoencoder, right? So it's like you're making this minimal representation here, but we're also adding in some other objectives. Then we can reconstruct this drug. And now we can do the gradient ascent we were talking about, because we can then design drugs that maximize this affinity function. So we talked about the contrastive learning objectives here. We're really caring about this vector space we are creating in the middle. But then there are some important parts about this, right? We also want it to be able to predict the affinity. So there are also some downstream parts of this network that we want to do well. We don't only care about this part, we also care about the second half. So we have the canonical mean squared error loss, Pearson correlation, and the ability to reconstruct the new drug from the vector representation. So we've got those parts of it as well. And then we basically run the training. So to give you a little bit of insight on how we did this training personally, is we used a data set called the Therapeutic Data Commons, which Professor Zitnik over at Harvard has developed. And what they've done for us, and this is also a very key point on model training here, they've got 180,000 drug protein pairs from 2013 to 2018 with their patents. Well, we got the patents, but not initially included in the data set, in 2019 to 2022 with 34,000 drugs. What's very unique, and I want to emphasize about this data set, is that a lot of times in machine learning, what we tend to do is we got 200,000 drug protein pairs. Let's make a training and testing set from that. So what we'll say is 80% randomly become our training set, 20% randomly becoming our testing set. Now, the problem with this is that a lot of the same proteins are going to be in your testing set as what's in your training set. So you're going to have 500 proteins, each with their pairs. You're probably going to have some overlap. And what that means is your model is going to learn, you really are not testing your model for out-of-domain generalization. So what we've done here, what the Therapeutic Data Commons has done, has made sure that none of the drugs here are in this batch, and none of the proteins here are in this batch. And that 100% guarantees to us that we are actually measuring out-of-domain generalization, not just, oh, can we learn it for these select few proteins, these select few drugs? And that's something that I would keep in mind for any model development, right? How good is your testing set? How contaminated is it? And benchmarking is another important thing, right? Our model does quite well compared to even the model that I was sharing with you earlier, and we're able to achieve state-of-the-art performance in terms of predicting the affinity. Now, how does that decoder do? Remember that decoder that we were talking about that can actually go back and make the real structure? Well, you can see for yourself right here. This is the original molecule, and this is the reconstructed one. And if you actually sort of flip that first molecule, it looks very similar, except for this one oxygen with that carbon here is just missing here. But other than that, it's very similar. And we're able to get a high performance in sort of figuring out the initial molecule. So we can do some gradient ascent with that. And the last thing is, remember, this comes back to contrastive learning. The reason why I'm actually sharing this is because of the patents, right? The patents were a major place where we did the contrastive learning. We wanted the patents to be in the middle of the drug and protein. So how well does that actually work? So if we look at the testing set, let's imagine three columns in a spreadsheet. The first column is the protein. The second column is your drug. The third column is the patent text associated with that protein and drug. If we use our model, convert each of these into a vector representation, so protein, drug, patent, what should we see? Well, we should see this patent lies in the middle of each. So now what we can do is the ideal middle, so the ideal midpoint is just the average of the first two columns, right? Are you guys following? Yep. Okay. Then the next thing is the patent. We want to figure out how close that midpoint is to the patent's actual position. So we can do that. Then what we can say is, let's shuffle the third column. Let's say all of that is wrong. Let's shuffle it. And we should ideally see that they no longer align properly, right? If the protein is far away, it should not align. And this is a different set of patents. And what we found is that is very much true. So the midpoint aligns properly on the aligned set. So my text is covering it, but it says aligned here, and that says shuffled. And what you can see is that the aligned show a much bigger cosine similarity to the midpoint than the shuffled. And that just shows you how well contrastive learning can generalize to understand now the meaning of text in the space of proteins and drugs. So we've created a vector space that has proteins, drugs, and text all in one. Now the next problem comes in, it's sort of interpreting the latent space. And we're going to go through the how of this in just a few slides. I keep referring back to 2D, 3D, but we'll get to that in a second. But the essentiality is this. We have 11,000 post-phase 1 clinical trial drugs here. And we processed all of them through our model. And the positions of each of these drugs is derived from our model. And what we can see is that they form islands, where each of these islands has a common denominator. That common denominator is that all of these molecules, or most of these molecules in these islands, are actually binding the same drug, or it's binding the same protein. And that shows us that the contrastive learning part is actually working, because drugs that are binding to the same protein are in the same region. And now we can see some of this in experimental screens as well. So what we did is we went to something called the Journal of Medicinal Chemistry, which publishes a lot of therapeutic screens. So they took around 40 compounds and tested them for an AD target called LMP2. And then they gave clearly the affinity values. And what you can see is that our predicted affinity aligns very well. But another thing that you can see, and I would ask you to observe, is that if you put a line smack dab in the middle here, you can see that the bottom half are the lower performing drugs, the top half are the higher performing drugs. And that just symbolizes to you how the vector space has sort of learned these important relationships. Yeah. Do you have a question?
So how is by where is it
by Kannan. So these are ground truth labels, yes. So like I'll actually go through how I did this analysis. So I took KNNs of this 11,000 space, so you'll get 11,000 KNNs, right? Then what we can ask is, well, let's arbitrarily select a few KNNs because you can't show all the KNNs. Then once we selected a KNN, we have the IDs of the drugs that are in that island or location, in that KNN cluster. And then we can ask what are the targets associated with them, and the majority target, so over 60% of the drugs were inhibiting vitamin K reductase proteins, or those family of proteins.
So the circles are the experimental measure.
Yeah.
The gradient are the predictions from the model. And you can see that the gradient matches the circle.
Yeah.
All of the red points, the red circles are experimental measurements right on the red, Does it not matter a lot if you have this not that simple?
Oh, here? Yeah, it's hard to see the separation with just 40 dots. But in principle, you can see it's a little more clustered to the right. You'll see this more if you have like 200 or 300 drugs. You can see the clustering a little better. But what you're able to see essentially is that there's like a line here where you can see all the points here are very much red and all the points here are very much blue. There are better ways of seeing it when you have more points. But yeah, essentially what we wanted to see here is that one, the embeddings do well, and two, the vector space is a good approximation. And two, we are able to predict affinity with high accuracy. And then I think this here conveys it a lot better. We did it with 121,000 drugs, and these are all experimentally tested. And what you can see is they once again form islands, right? And these islands sort of share, well, all of these drugs in this half of the UMAP, or rather, I would even say this place right here, are the ones that actually bind positively to EGFR. And once again, I'd like to remind you that these are all drugs. We embedded them using this model. And then what we're seeing is that the drugs here are actually the ones that
Continue, sorry.
are the ones that are ones that bind to that protein.
Yeah.
Yes. No, these are not ones we trained on.
Okay.
Yeah. These are the ones in PubChem. So PubChem, for every single protein, they'll have a list of drugs that they've tested. EGFR is not one of none of the drugs are in our training set. And what we're able to see through that is that all the ones that are positive hits are in this half, or rather, very much the density is higher here. So then a few weeks ago, we were asking the question, well, if these are, we're seeing there's some more islands that are developing, right? Yeah, it was Sunday. We were asking the question, well, if they're forming islands, do they have unique sort of, do they bind to different regions? Why are they in different places? So you learned about docking a few lectures ago, and what we did docking with 50 from each, because you might have also learned that docking is very computationally expensive, right? So we took 50 from each of these islands, we topologically sampled them, and then did 50 from each one. And then we plotted those, right? There are a couple things we noticed from this immediately. First is we can't immediately trust these results because docking is inherently has, it's sort of probably has error. And two, the problem is that it is susceptible to like things like molecular weight or things like that. So don't trust this analysis yet, but we do have some like initial hypotheses that, well, each of these sort of islands are docking differently to this protein. And you can see that more with greater clarity in this bar graph than here. But what you can see is that certain clusters go to different coordinates. And again, I do want to put this disclaimer that it's filled with error, for starters. And two, it could just be because the molecular weight of these compounds are much higher that they could be in a different location. So there's much more work to be done. But the idea is that we can go from these latent space representations directly to understanding their structural implications using tools like docking. Now, we've looked at a case study of sort of using contrastive learning, using these representations in practice. But now we want to go a little bit farther. We've got, I've kept on showing you these figures where I've shown you 11,000, 121,000, all these protein, all these drugs. And now our question is, how do we actually use these embeddings, right? At the end of the day, these are what, here I've shown you a 7-dimensional vector, but in reality, these are thousands of dimensions. And as you saw from our model, it's 1,024 dimensions. How do we actually use them? So there are two key aspects that you might have learned, and that's PCA and UMAP. And they're used for entirely different, their techniques they use are entirely different. PCA sort of finds out, well, if you have a large swaths of data, it sort of tries to identify which axes have the greatest variation. And using that, we're able to sort of approximate the 2-dimensional and 3-dimensional coordinates of where we as humans can see them. And it actually does a great job of approximating these clusters. Now, UMAP does this a little bit differently. One of your classmates here was talking about the KNNs, or K-nearest neighbors. So we can sort of look at large swaths of data again and group them into K-nearest neighbors. Then using this K-nearest neighbors and sort of looking at the distances between them, we can create neighborhoods. And you can define parameters for all of these that I'm talking about. But in essence, it allows us to create a way that we as humans can visualize n-dimensional data as approximating them by just looking at their neighbors.
I want to jump in a little bit. So first of all, sorry for all the interruptions. First, it ran out of space. It wasn't recording anymore. And then I was like, oh, I'm on the wrong network, so the quality won't be good anymore. And I was making sure there was a microphone. Anyway, so I want to add a couple of things. So basically, principal component analysis is, of course, a linear dimensionality reduction technique. And it is a true space. It's a true dimensionality. In other words, if you have an x-axis and a y-axis and a z-axis and, whatever fourth axis, this is the actual, like a point on that axis means one thing and one thing only, which is the amount along that dimension of the variation that you go down. The problem with PCA is that each principal component captures a very small fraction of the variance, especially when you go to 1,000-dimensional spaces. The first PC will capture, I don't know, 5% of the variance if you're lucky. And then the next one, 3% of the variance if you're lucky. And then it sort of diminishing returns. So that basically means that even with PC1 and PC2, you're going to capture a very small fraction of the variance. So yes, you want to visualize something that is a true representation, a true space. And it would be lovely if 2 dimensions captured everything. But in these super high dimensional objects, 2 dimensions don't really begin to capture the true essence. You can think of UMAP and T-statistics, stochastic neighbor embedding, TSNE. You can think of them as effectively capturing, let's say, 40 dimensions, not 1,000, but like 40, but like warped in a way that they squeeze onto a 2-dimensional map. Okay? So it's not a dimensionality, of course. And some people are out there are basically going to go crazy if they hear you're using a UMAP. But like, the whole point is that if you understand the mathematics behind it, if you understand the limitations, it is an extraordinarily practical tool. And I want you all to sort of recognize that this is not a real space. You're not going to move down it and think that you're moving down one particular dimension. No, think of it as basically taking all of the dimensions in an Escher painting or in Alice in Wonderland and just like laying them out flat. And sometimes when you're going to be moving along this direction, you're going to be going down some big highway. And then other times you're going to be going down a road that actually goes in a convoluted way. And other times you're going to be going down, yet another path. So basically every part of that super high dimensional space is projected, every dimension is projected into some parts of the 2-dimensional space. But in different regions, you're obeying different coordinates and different axes. Does that make sense? So variation is absolutely captured in a UMAP. And as you're moving down some of these axes, you're moving down a gradient in one of the latent dimensions. Sometimes it's going to be dimension 13, other times it's going to be dimension 7, other times it's going to be dimension 2. And even if you take the same line and you move it this way, you're going to be shifting along different dimensions. But the whole point is that it flattens it out so that we, measly humans, can interpret it. Make sense?
So yeah, that it's vital to sort of understand the how behind these dimensionality reduction techniques, because we're going from n dimensions to 2 dimensions, and it's important to capture the variation and important parts of this manifold. Now, one of the other parts of our lab that we've been working on is sort of, we've got n-dimensional spaces, we can convert them to 2 dimensions using these techniques that Professor Kellis had just mentioned. How do you actually interact with them as humans, right? Implicitly, they capture, as Professor Kellis was mentioning, these roadways and highways. And because they capture that, we can do some mapping, right? They are real maps. You want me to?
Sorry, I realize I'm not yet logged on.
Yeah.
But carry on.
No, I was going to talk about this anyway. So
no, go ahead.
So essentially, what we want to do is sort of figure out, well, can we actually map these sort of dimensions? And our first attempt at that is mantis, which sort of allows us to work with these hyperdimensional spaces in a human interactable way. So what we looked at in the previous slide is a way of visualizing, and now what we're saying is interacting. So there's a difference, right? So let's go ahead and
So you go back to the previous, oh, sorry,
tab.
Control Shift tab.
Control Shift. Okay.
Oh, lovely. This is the downside of a live demo on a computer that I haven't used in a while. So let's see if this will work. Yay, it is working. Is it loading? Oh my God, it's loading. Yay.
Yeah.
Right.
So essentially what we are looking at here is that same 11,000 space that we were seeing in the last few slides, right? I kept referring back to this 11,000 drugs that are all phase 1 clinical trials. These are all of the drugs that have passed phase 1 clinical trials. But this time I can move around. I can sort of zoom into a specific cluster if I think that might be of importance, and then ask questions about this, right? And the idea is that we want to now interact with it. So I'm going to pull up this live image preview, which allows us to look at the chemical structures. And we're going to try to look and find out if there are structural motifs that sort of unify a specific cluster. So for example, let's go to this remote island here. Like it's so far away. What sort of makes that
They only, can we turn the front lights off?
Yeah, let me turn it.
Oh yeah, maybe the
I've
dark
been
mode
trying
might
the
not
front
be.
light, but it hasn't been working, so.
Okay,
That's what I'm doing, but this is,
so yeah, maybe dark mode was not the right the right thing for this, but essentially, okay, I want you to. I'm gonna move my cursor, and it's gonna show you the.
Let me make that window a little bit.
Yeah. It's going to show you the chemical structure here. You don't need a lot of chemical structure knowledge to even look at this, but what I want you to just observe, right, is that all of these chemical structures are one big. They have a lot of double bonded oxygens, right? And there's something that unifies them, right? And these are captured. One, we can get the visualization, the 2D, right? The next thing is that this latent space, because of the way we designed it, captures function, right? It captures where and which proteins these drugs are going to bind. So this island of molecules is likely going to bind to the same protein, right? And we can do the same for this island here. And you can see some very similar drug structures. And we can do the same for different islands. And as we go along, we can ask questions. Well, maybe this island of drugs is the ones that we should be testing for this particular protein target that we've been working on. And the idea is that interacting with embedding spaces might allow us to uncover more insights than just using them in a downstream machine learning pipeline. The representations themselves have learned so much that if there's a way that we can interact with them, then maybe we can uncover some new insights. Professor Kellis, do you want to think? Sort of
I just wanted to keep browsing. This
This is a lot of fun.
Yeah.
So no, I mean, I think this is so exciting. In other words, basically being able to
Wait,
walk.
that place is all like can see all ions and elements. So you can see like it's yeah.
So I mean, I'm a data geek, so like being able to sort of like spend time understanding chemical structure that way is just so exciting. Can you tell us a little bit more about the tree or any of these things? Are they meaningful?
Here, I think the tree is, yes, maybe. Cannot be with this one. Yeah, no.
It's not on?
No, it's not.
Let me see if the Explorer is
It won't be maybe
Okay.
on this. No. So in other molecular spaces, what we've been able to do is sort of say, well, you remember that you can see how the structures in these islands are very similar, right? What we wanted to do is, okay, maybe there's a common denominator, like quite literally a common denominator that sort of encompasses all of these structures. So there's a way to sort of put stars, like, you know, that Spiles representation. So we keep a scaffold and we say these parts are empty, but they all follow the same general syntax. And the farther and farther you zoom in, the more specific the Smile syntax becomes. So that's what the tree usually shows if you're in a different molecular space. But this one.
And then what about the actual function? Is that something that I can get some insights about?
Not yet. But yes, eventually the idea is that we can color. Right now these are colored by cluster. But in the future, we're going to color them by function. We're going to color them by the proteins that they bind to, which we can predict through our model, right? And the idea is that if you just select maybe EGFR, we can figure out all of the molecules that bind to EGFR with high strength. And doing this allows pharmaceutical companies, researchers to sort of understand the space of chemistry, right? I keep talking about that space earlier, and this is why it mattered so much, that we can assemble this sort of.
So anyway, for those of you interested in chemical structure,
like you should just spend some time exploring the space.
Yeah, it's quite. You can if you're curious, you can just go into wild deep dives in these islands.
The only 11,000 post-phase pharmaceutical drugs.
Yep, these are actually, well, not all of them are being used, but a subset of 3,000 of them are actually in the market being used, like for patients right now. Those are all the FDA approved drugs. So
Are the patents also part of this?
not in this one, no.
Okay.
Control shift. Yeah.
How big is the model and how long will it take to train?
Yeah, that's a great question. So it's actually a very small model for comparative purposes, only like 43 million parameters, and it's like, to put that into comparison, like usually ESM is in the scale of billions, so like we're very small, and the training time takes usually under a day, because that's the benefit of foundational representations. The heavy weight of it goes to the big companies like Meta who have the power to do them. And now we can just sort of extrapolate and make these joint representations that are more meaningful, that can capture all of this important information. Control Shift Tab?
Just up top.
Alt Tab, okay. All right. So the idea behind these sort of interactive visualizations is you can explore a region, maybe make a hypothesis, and then eventually make some new findings, right? That's the idea. And this is all, again, from our model, these latent representations. Okay, now to the fourth part of today. We've talked a lot about data in high dimensions. The problem is, okay, we can interact with them. We just saw how we can interact with them in 2 dimensions and 3 dimensions. How do we interact? What is a better way to interact with them? Maybe we let AI interact with AI, right? Like in the sense that maybe we as humans can't fully understand these n-dimensional representations, but maybe artificial intelligence can. And I don't mean this in the way of downstream tasks, right? You could easily take this latent representation and train downstream models. But instead, is there a way to interact with these high dimensional spaces using agentic frameworks? So now there's a lot of like nomenclature around the word agents, but to put it very simply, let's say you ask a question to an agent. An agent, you can simply call it an LLM. We'll just say it's an LLM with tools. So an LLM can be anything from ChatGPT to Gemini to Meta's Llama, and then you can have tools. And these tools are the medium with which they will interact on these n-dimensional spaces. These tools can be arbitrarily anything. And I think in your piece set, you're going to be seeing tools like anything that can be defined as a Python function is a tool that an LLM can use. And that's the idea behind agentic thinking, right? So if you give a question, this agent will use tools, get a result from that tool. So a tool could be AlphaFold, for example. A tool could be ESM. And the agent would receive the response from that tool, come up with a response to answer your question. So where does all that n-dimensional stuff come in? Well, these tools are a way to interact with these n-dimensional spaces. So you're going to see some exposure to these Python tools and how to do this in your piece set. But how can we use these agents with these n-dimensional representations to do drug discovery? That's what we're going to look at now. So what we've been doing, or a process that you can use to do agentic drug discovery, we're going to look at target selection. So we're going to veer off a little bit from the therapeutics, but then come right back to it. But essentially what we've got here is at the beginning, we were talking about scRNA-seq data, the single-cell RNA sequencing data. We talked about biobank data. We talked about all of these data modalities, which are in essence n-dimensional vectors, right? You could also use the same representations. So then what we look is these agents are going to work with these data to give us a hypothesis, right? So let's imagine we have Alzheimer's disease and we want to find a drug for Alzheimer's. In the next 2 slides, that's sort of the prompt that we're going to be going for. So we have single-cell RNA sequencing for this. We have differential expression information for this. We're all familiar with those terminology, right? Okay. And then we have the biomedical literature, all that from PubMed. And we also know what genes express themselves at the same time. So these are the input data sources. Then we have this agent, give it tools. So tools could be like read literature or find papers about and then a specific term. Or is this gene overexpressed? And then it can basically access our gene expression data. And then what it can do is it can sort of interface with this. Instead of humans asking questions to the data that we were just looking at, agents can start asking questions to the data. So it'll say, oh, I'm going to start by looking at the 20 to 30 of the most consistently implicated proteins. So it's going to ask, what are the most differentially expressed genes in Alzheimer's, which was taken from scRNA profiles, right? Then we can see several targets have these strong support. So then we can look at the 5 top differentially expressed, and it'll look at the totality of evidence, right? We've also got PDB structure, all of this information. Then we go to the validation agent. So what's also important for a protein target for a given small molecule therapeutics is it needs to be non-toxic. So if I give a drug that attacks a specific protein, we want to make sure that that protein is not overexpressed in other cells as well. For example, that protein is very expressed in your heart. Well, we don't want to send a drug to that protein, right? So what we've done is we've also captured expressions per organ, and we gave that information to the agent as well. And what we wanted to do is make sure that the expression is very specific to the organ that we're going for. So in this case, the brain. If it's not, we're not doing that target. And then we get a ranked list of targets. Now we're going to circle right back around and go back to the therapeutics, right? And this is where the affinity net and sort of the landscapes that we were talking about come back in. So we have a selection agent that sort of evaluates these molecules. So we have a way of sort of getting the top 10 molecules for a specific protein target just by evaluating the ranked affinity, right? Not too complicated there. Then we can have this agent then look through different parameters, right? You've learned about different parameters for these molecules, right? The drug likeness, BBB permeability, all of this important information exists. And now these agents will sort of evaluate each of these criteria, then prepared a ranked list. So let's come back and revise what we did. We found the targets, we used our models, we used these landscapes, we used all of these databases, and then finally came up with a ranked list of drugs just by going from single-cell RNA sequencing and the disease that we wanted to stop. So the takeaway here, once again, is that agents can help us navigate these large swaths of data. So you can have representations, we can have agents that allow us to navigate these representations. So here's a case study where, again, it's a continuation, where we allow this agent to basically identify drugs for Alzheimer's disease. And what it's doing is it's first reading through the literature, it's looking at the differential expression. So if you can read through this, and then, okay, so so the first thing is like, I will begin by searching for cell type specific mechanisms implicated in Alzheimer's. I do want to note it's actually very amusing to read these agent transcripts because it's like it's literally thinking out loud. And I think in your piece that you might have a very similar experience. It's just literally thinking out loud. So it's using this tool, so that's highlighted in blue, searching corpus. Literature indicates that inhibiting GSK3 beta could reduce tau pathology.
I just want to add a couple of things. So basically, agents are like us. They are creative. They are persistent. Something will bug and then they'll try again, or they'll try a different way, etc. This is wonderful if they're doing something that you like. If they're doing something you don't like, you call it hallucination and you sort of bash them. But they are in many ways very similar to sort of how humans think, in creative, flexible ways. Tools is what makes us, frankly, function well. If we had to sort of multiply 7-digit numbers all the time, we'd suck. And people would be like, oh, look, these humans are hallucinating. I'm like, no, but we know how to use a calculator. And it's the same way. So basically, you want to combine in your PSETs, in your projects, in your own research, you want to combine the creativity and the flexibility and the resilience, the resourcefulness and the resilience of agents with the hardcore, boring, dry, repetitive, reproducible behavior of tools. And that's what the gray boxes are here. So the agents are basically thinking in the blue boxes, and the tools are executing in the gray boxes. And the agents are like you and I can interpret a 7-digit multiplication because we know what it did. We have a model for what happened behind that box. And so do the agents. So the agents they're terrible at multiplication. They suck. But that's not what they were made for. They were made for using calculators, kind of like we were. Okay? So basically, that's why the different colors here. So the blue are the agentic calls, and the gray are the tool calls, and the green are the results, and the pink are sort of the final result of that column, if you wish.
Yeah. What I might add is that LLMs, they have debatable purposes, right? We've got modeling, right? We've got machine learning. We can find these representations. These sort of LLM-based systems are used to integrate information. So that used to be a very big pitfall. How do we integrate sources? This is how you can do that, exploiting this niche power of decision-making that LLMs have. And we actually found some candidate molecules here, and they're being tested, have been tested, and we're working on sort of looking at that information.
But what was the pipeline?
Yeah. So what we see is that it's checking differential expression. So some of the tools that we talked about in the last slide. Then after that, it's identified 2 target genes, GSK3 beta and PTK2B. Then we want to figure out the validation. Remember, we talked about the toxicity of these targets. We wanted to make sure that they're not toxic. There are 2 ways that we need to do that. First, well, this is less related to toxicity, but we don't, if we're finding a combination of drugs, so let's say we're allowed to give 2 drugs to the patient, we don't want both of the drugs to be attacking the same protein or the same same pathway of proteins, because that's redundant, useless. We need to figure out a better way, right? So that's what get modules does. So if the expressions of 2 proteins are very correlated, they're likely in the same pathway. So what we say is we get the module, these targets are not pathway redundant. So that's exactly what I was saying. And this is the transcript of the agents. And what you're seeing is that then it's checking for tissue expression. And precisely what I was saying in the last slide, right? GSK3 beta has high heart muscle expression, which might not be favorable. Then the next thing is it's reading the literature, making sure to find any biomarkers that we could exploit to see if the drug is working. Then we use our landscapes. We find small molecules, we do the docking. So we can actually integrate our model, we can integrate docking, we can integrate literature. And you can see all of these information just coming together in one place as if a human is operating all of these models. And that's sort of the idea. And then it gives us a nice report to make sure that we can capture any hallucinations or any incorrect tool calls that it has made. So here's a fun little demo of like the agents literally working like humans do once again, the black might not have been a great choice. It's very small text, but you can see it's like typing. So you can see here it's reporting and generating the report. Here you can see it's selecting the drug and it's going through each drug and checking them. Here it's searching through PubMed. Here it's selecting the target. And you can see there's varying levels of thinking that are required for each stage, right? For the target validation and the target selection, that requires a lot of thinking. It needs to search through literature. It needs to do a lot of decision making. Drug selection is a lot more dependent on the tools. Like we give it the tools, you're just putting them together. The report generation obviously just captures all 3. Okay, so we'll go to the next slide. Let's see, I think it's about fine tuning now. All right, yeah. So we have about 7 more minutes. So the important part about these agents is they've been trained to do a very specific task. Find the next word, right? ChatGPT was taught that. All of these large language models can just be to think of the next best word. So the problem is they might not be so well versed at using my model, or it might not be so well versed at doing docking, right? We have to sort of teach the model to do that. One way you could do that is sort of saying the prompt, this is how you do it, this is how you do it, basically like going into the mind of this person and just basically telling it all of this. But every single time you use the agent, you need to basically do this.
The prompt window itself, the context window, might get saturated if you have too many instructions every single time as well.
Exactly. And that can be very, that's very inefficient because you want to use the context window for more important stuff, right? You want to use it for the scRNA sequencing data or the results of all of that. So how do we fix this problem? Well, once again, OpenAI and a lot of other companies and a lot of other foundations have sort of figured out the way to sort of make these LLMs become practical tools. And the way we can do that is by setting up problems. And inherently, if you ask ChatGPT the same thing twice, it's not going to give you the same answer. They're stochastic. And we can use that stochasticity as a way to sort of rein in these agents. So what we say is, the way we can sort of fine tune these agents is you sort of generate problems. In this case, it'll be like, we're gonna make it simple examples for today. But these are some of the tools that we're going to give it, lookup tools, send emails, or making documents. And we generate some problems like find customers who will be interested in my products, which businesses are my competitors. These are the literal prompts we're giving to this LLM. Then it generates multiple responses that might use a different set of tools. One response might use no tools at all. Do we prefer that? Absolutely not. But then one tool, one response B, because of the stochasticity of these models, might generate the perfect response. And we basically say response B got 0.9, response A got 0.3. There are different ways of evaluating this. We can have another LLM basically evaluate an LLM. You could have an answer checker, like a base, like you could do this several ways, right? So then what we can say is, well, we basically tune this model repeatedly, just like you would do gradient descent. This is just basically reinforcement learning. Then instead of fine-tuning this massive 5 billion parameter LLM, you're just creating a model adapter that just sits at the end. It's a low rank matrix that just gets added to the end. Then you deploy the model with this new adapter that just clicks onto at the bottom of the model. And what that allows us to do is now it's learned these tools. It's learned how to use the lookup tool, the send emails tool, and the make documents tool. And in our case, it's learned how to use docking, learned how to use affinitynet, learned how to use scRNA sequencing data a lot better now that we've explicitly taught it or said what we expect of this model. And the idea is that reinforcement, there's another way that we could go about the same problem, and that's saying, well, this is exactly how a human would go about this problem. And you would do this like 15 times, right? You would need to generate swaths of data of humans approaching this. One, that's logistically very difficult. Like if you have to generate that much data. And two, humans aren't perfect too. And what we've found repeatedly is that this type of reinforcement learning, this is SFT, that's called supervised fine-tuning, the approach that I was just talking about. It's way less effective than PPO, which is preference optimization. And the RL here performs much better at sort of responding. And this is a very early version of ChatGPT, and you can see it's like 2022, even before it was released. What they found is you have to do reinforcement learning tuning after the model has been created to even learn some of the most important parts of a model. So Professor Alm, when we discussed, had told me that we had sort of looked at this 4 steps of drug development, right? You go target identification, compound discovery, lead screening, then lead optimization. You start with sort of what protein should I target? Then you go through the big space of compounds, which is the whole topic of today, right? How do we go through all this genetic therapeutic data? Then we find a candidate library, screen every single one of them to figure out which one works, find one that works and sort of modulate it to make it better, and then hopefully it works, and you send it to patients, right? And that's essentially what we talked about in disguise. And to sort of show you how that actually plays out, right here, this agentic drug design step sort of puts everything together. It's target identification and compound selection. So what happens here is that the target is identified through this agentic process right here. Then you have the compound discovery happening through the machine learning models. This agentic stuff is basically at the center of this, but the compound discovery, or like the compound selection steps, are happening through the joint representation learning, now that we've learned to map between proteins and drugs. And then the next step is the lead screening and optimization. Maybe in the future in this class, we could see that lead screening and optimization could be done using a machine learning model. It could very likely be done even today. But in most cases, that's being done at this level, the experimental level. So once we get a few great molecules through these steps, we go to the laboratory, then make modifications that might make it even better because we can get to the precision that we need to go to the next steps. So essentially what you're seeing in the last slide has made its way here. And the idea behind systems therapeutics is sort of approaching drug discovery through the angle of combining a variety of multimodal data to arrive at drugs that can actually save patients' lives.
And I just want to say that this space here is, yes, enormous. We basically have like 11,000 already, post phase 1 drugs, but it's a tiny subset of all chemistry that has already been screened somehow and in ChemBank. That's 121 million molecules. What fraction of chemical space is that? Epsilon, nothing. 121 million has like only like about 9 zeros. All of chemical space is on
To the power of 60.
Yeah, on 60 zeros after the 1. Okay, so basically, the 121 million drugs are a tiny fraction of all of chemical space. Why am I saying that? Because here, when you're actually doing your experimental screening, you can basically zoom in into a tiny little part, almost white. Kind of like, the James Webb Telescope basically looks at dark parts of the sky and zooms in and like turns out they're full of galaxies. Okay? So in the same way, you can basically take a completely blank part of chemical space and then zoom in, and it turns out there's still more stuff there. And that's where the high throughput experimental testing comes in. So basically, we have a computational lab, but we also have an experimental lab. And our experimental team is basically currently screening drugs, both in organoids and in cell cultures, and we're basically screening additional compositions of matter that we're making in collaboration with the chemistry department in Brian Petuluta's lab to basically synthesize new chemistry to go and explore those like white spaces, the place where there's no drugs yet. Because our model cannot be possibly expected to capture the fine-grained grooves of that latent landscape of binding affinity in those spaces. Saathvik has shown that a lot of the space is continuous, at least to some resolution. So basically, we're hoping that if you continue sort of digging in there, you might actually find additional drugs. And then you can better model that part of the landscape and then feed it back into your models. Because it is ultimately the seeds from the 11,000 drugs and from the, ChemBank and PDB and the Therapeutic Data Commons and all of these things that sort of allowed us to even train the model that then gets a sense of the landscape, but some parts are just still unexplored. So that's where the active learning and the sort of relative feedback comes in. Who feels that they've learned something? Yes? Give Saathvik a big round of. What I did not mention at the beginning is that Professor Saathvik is actually, 18 years old?
No, I'm going to turn 20 in a few.
He's 19 years old. So he basically joined my lab as a freshman with 9 patents, 7 publications, and 700 citations. So I take no credit for his awesomeness, but he's basically developing all of that stuff as a first-year undergraduate. Okay, just to be clear. So I see a bunch of mouths just dropped. So I just want to say I'm incredibly proud of him, and this is like the vast majority of this is like his own work and doing. And I'm saying this not to intimidate you, but to tell you that this space is exploding. Now is the time to just dive right in. One of my best computational team members saw some of the work that they're doing in agentic workflows, and he's sort of reproducing all these tools by actually a 16-year-old student, Aditya. He basically said, Manolis, can I learn how to do experiments? Because I'm not going to compete with these guys. So I don't want you to have that reaction. On the contrary, I want you to have the reaction of, whoa, this is amazing. I can do so much more. And I want to incorporate these capabilities, these tools, these embedding space, cartography and navigation, this interactivity that Mantis enables, all of the agentic pipelines that Sphinx and Mantis and all of these other tools enable to basically do what we as humans should be doing. We shouldn't be like browsing YouTube videos or whatever the latest fad is. We should be changing human health for the better. We should be changing climate. We should be like fixing our planet. We should be saving humanity, like ending disease, etc. So it's not like, oh, it's going to take our jobs. No, we need 1,000 times more scientists today than we have. And those agents can help us reach the productivity that, frankly, we as a human race deserve and need in the service of humanity. So no pressure, but each of you should be like 100 times more productive because there's so few scientists in the world. And I'm hoping that these agents are allowing you to just dramatically boost your creativity, that all of these robotic work, because we're not going to cure Alzheimer's in the next 6 months and then run out of stuff to do. There's so much more science to be done. Everybody excited? All right, awesome. Thank you again.
Thank you.
And again, Saathvik, you already know because he's a senior mentor in many of your projects, despite of his young age. Anyway, super proud of it.
Thank you.